Today we have the distinct pleasure of hearing from Dr. Samuel H. Sternberg, Assistant Professor in the Department of Biochemistry and Molecular Biophysics at Columbia University. Dr. Sternberg received his PhD and postdoctoral degree from the University of California, Berkeley, where he studied the mechanism and engineering of CRISPR-associated endonucleases in the Doudna lab. He is the recipient of graduate student fellowships from the National Science Foundation and the Department of Defense, and was awarded the Scaringe Young Scientist Award from the RNA Society and the Harold Weintraub Graduate Student Award from the Fred Hutchinson Cancer Research Institute. Dr. Sternberg's current research focuses on using biochemistry, structural biology, biophysics, and genetics to further explore the function of CRISPR-Cas systems. He is the author of numerous high-profile scientific publications in high-impact academic journals, as well as the co-author of the New York Times best-selling book, A Crack in Creation, Gene Editing and the Unthinkable Power to Control Evolution, which details the history of gene editing and narrates the dramatic events leading up to and following the discovery of CRISPR-Cas9 gene editing technology. He is considered a leading expert in CRISPR technology and is invited to speak internationally about his groundbreaking research, as well as the ethical issues surrounding genome editing of human cells. Please join me in welcoming Dr. Sternberg.
Thanks, Hannah. I think it's the first time I ever heard you call me Dr. Sternberg. So we're friends from college, and hopefully I don't embarrass you today. So, or I'll try to, yeah. So thanks for the invitation. It's a real pleasure to speak to an audience I don't typically speak to. Brief disclosure, I have a little bit of stock in a couple companies that develop CRISPR technologies for human therapeutics. And I just wanted to also make clear that I'm not a physician. I don't see patients. I'm not actually developing medicines in my own laboratory. My background is really in basic research and in the basic biology and technology development of CRISPR. So as a researcher, I think biology has really transformed itself in the last 10 years with the advent of a facile new approach for precisely rewriting genetic information in living cells. This has been on the covers of all the top magazines. You can see in the middle this kind of exponential growth in publications using CRISPR as a method. There have been dozens of companies that have either formed around new CRISPR technologies or adopted CRISPR into their main platforms. Some of you may have seen CRISPR pop up in the X-Files or Hollywood movies like Rampage. And CRISPR's, of course, also made it into very mainstream magazines like The Economist or The New York Post because of some of the more controversial sides of gene editing, namely this potential to use tools like CRISPR in the context of human reproduction. And so although this is also not an area in which I work, I'm going to talk a little bit towards the end of my talk about some of these issues that I think as scientists and physicians we really need to be thinking about because, of course, there are many different ramifications of the work we do. So one of my goals today will be to really deconstruct what is CRISPR. So I'm going to start really with where this technology actually came from, how it's being used today in a number of different research areas, some of the ongoing challenges in the full development of these tools. Again, some of the questions around germline gene editing, and then I'll end by giving just a brief snapshot of some of the work we're doing in my lab, which focuses on developing new kinds of tools by going back to the basic biology. Actually, I realized I wanted to use my laser pointer. So one thing I want you to keep really in mind today is that CRISPR exists in these two worlds. Probably many of you have heard mostly about CRISPR in the context of gene editing, but CRISPR is something that exists in nature. About half of all bacteria on Earth have CRISPR in their own genome, and CRISPR is something that evolved to protect bacteria against viruses. So I'm going to spend the first 15 minutes of my talk or so giving the backstory on this world of CRISPR biology, which goes back to 1987, and then move into how basic discoveries in bacteria led to this technology development. So 1987, this is the first study that described what we now call CRISPR. It's a pretty esoteric publication from a Japanese group that was studying genes. They were doing old school gene sequencing So this entire paper was a single gene sequence from a metabolic gene. But what they discovered in the last, they mentioned in the last paragraph of the paper, was what they called an unusual structure in the region flanking this gene that they were really interested in. This structure had 5 homologous sequences of 29 nucleotides arranged as direct repeats. So this was a little bit surprising because this type of periodic array hadn't ever really been seen before. So they ended their paper commenting that the biological significance of these sequences wasn't known, and actually that would remain a mystery for about 20 more years. So in my lab, we spent a lot of time looking at genetic information, so we can actually go into the genome of E. coli or many different bacteria and look at what these unusual structures look like. And here, what I'm highlighting yellow is this periodic pattern that was first uncovered by those Japanese researchers. And you can see this beautiful periodicity shows up as these diagonal lines on the page. And what was even more interesting is by the early 2000s, it wasn't just E. coli or Streptococcus thermophilus. It was about half of every bacterium that had a sequenced genome had a structure that looked exactly like this. Not the exact same sequence, but the same repeating pattern. And so it was in 2002 that an acronym was finally coined to describe these structures. I will say there were many acronyms before CRISPR, but I think CRISPR is the best one, and it was the one that we finally agreed on. And it really describes this pattern. They're clustered, regularly spaced, short, palindromic repeats. But still, the observation that these are widespread in nature didn't tell us what they're actually doing. The first clues to that came in 2005 when 3 groups made kind of one of the most interesting discoveries, which is if you ignore the repeats for the moment and focus on the little snippets of DNA spliced in between the repeats, these were often perfect matches to viral genomes. This was the first clue that perhaps CRISPR has something to do with antiviral immunity. So brief refresher from maybe intro genetics. These are not human viruses, these are bacterial viruses, which are the most abundant form of life on Earth. They outnumber even bacteria about 10 to 1. They can be found in really any environmental niche where bacteria are present. Here are real electron microscopy images of, in this case, a cell being infected by a number of different bacteriophages or viruses. This is another micrograph. You can appreciate this kind of capsid structure where the genome is stored, and then these tail fibers that help guide the attachment onto the outer cell surface. If you look at a simple cross-section, again, these tails have proteins that can bind to receptors on the outer cell surface, and then there are pumps at the base of the phage that actually inject the DNA inside the cell. So this injection is often a fatal event for the bacterium. If the genome goes undetected, it will rapidly turn on gene expression, which produces more viral proteins, guiding the production of additional capsids that eventually burst the cell from a buildup of pressure. So this model system has been studied for over half a century. We've known since the 1950s and 60s that bacteria encode other types of immune systems, such as restriction modification. You've probably all heard of, maybe used restriction enzymes to do cloning. So the question is CRISPR another type of immune system that bacteria possess to counteract this threat? So I think in one of the most beautiful sets of experiments, this was proven to be the case in 2007 by a yogurt company. This was a company studying Streptococcus thermophilus, which is the major workhorse ingredient to ferment milk into yogurt and many cheeses. It's a $40 billion industry, and these companies are growing huge vats of these cultures, and so a viral infection is a big time and money cost if you can't control it. So in the course of studying this bacterium and trying to evolve strains that are more viral resistant, they proved that indeed, any time that the cell had a particular sequence matching a virus that they tried to infect the cell with, those cells were completely immune. So you can really think of the CRISPR as functioning like a molecular vaccination card. Just like you might keep track of vaccinations your baby has received, bacteria are doing this within their own genome. They're keeping a track of past infections, in the form of these short snippets of DNA, and then using this information to recognize those repeat invaders during a future infection. So here's a slightly more technical slide to just show you what our current knowledge of CRISPR-Cas immune systems is like. Here's a way that in the literature you'll often see CRISPRs depicted, where the black diamonds represent the repeat sequences and the different colored squares are those short, virally derived spacer sequences. And a key feature of these immune systems is that these repetitive regions are almost always flanked by genes, CRISPR-associated or Cas genes, that encode proteins that work together with the CRISPR array. So in the first stage of this immune system, which is interesting in and of itself, although I won't speak more about it, small snippets of DNA from a virus need to be integrated or inserted in a directional fashion into the CRISPR locus. These loci are then transcribed initially as a long precursor RNA, which are then enzymatically chopped into short CRISPR RNAs or guide RNAs. These guide RNAs get bound by one or more Cas proteins, such as Cas9, and then effectively these complexes are surveilling the cell for the presence of any invader sequence that matches the sequence of the guide RNA. This match is identified through RNA-DNA base pairing, and once a target DNA is identified in such a manner, it gets targeted for rapid destruction. So the star player in this pathway, in the context of gene editing, although I'm going to tell you at the end that there are many star players, but for now, it's this enzyme called CRISPR-Cas9. So you'll see in the popular science literature, it's often called molecular scissors. It's a nuclease enzyme. It has two different nuclease domains that guide the cleavage or scission of both strands of DNA. In gray, this is a cryo-electron microscopy rendering of the Cas9 protein bound to the guide RNA in black, and here's it hybridized to a target DNA in red. So again, in a bacterial cell, Cas9 makes a DNA break, and this triggers the eventual degradation of the remaining viral genome. So the two properties that make this system so effective are A, it has this enzyme that's the arsenal that does the cutting, and then the guide RNA is really providing the GPS coordinates. And for those of you that are more familiar with programmable nucleases, you'll appreciate that Cas9 was not the first enzyme that was used for gene editing. In fact, there's a whole couple decades of literature on the development of enzymes to enable gene editing. But using a guide RNA makes this by far the most flexible and programmable system, where simply by changing the guide RNA, Cas9 can be rapidly deployed to a new sequence of interest. And by the way, if anyone has questions, I'd love to be interrupted if I'm not explaining something well. So here's a quick animation to kind of give you a sense for what this might look like in real time. So a cell is being infected by this bacteriophage, which latches onto the outer surface. The pumps are now injecting the genome inside the cell. And at this point, the cell has about 15 minutes to recognize and neutralize that DNA before the virus will replicate itself, and at that point, it's too late. So here's the guide RNA and Cas9 forming a co-complex. This is going to patrol the cell looking for a matching sequence. Again, that match is identified through the formation of RNA-DNA base pairs, which generates an R-loop structure. And at this point, the nuclease domains will cleave both strands of the DNA, triggering the eventual degradation of the rest of the genome. So that's summarizing a ton of literature. And that brought us to roughly 2011 or so, where the field now understood the fundamentals of how CRISPR immune systems work and what the molecular pieces are that are actually triggering this interference step. And at that point, the field of CRISPR really intersected with this pre-existing field of gene editing, because it was immediately recognized that having a programmable cutting enzyme would allow you to potentially repurpose that cutting function for introducing targeted double-strand breaks in other cell types. So that, again, led to this kind of transition from CRISPR biology, something that exists naturally in half of bacteria on Earth, to the development of molecular components taken from bacteria to be repurposed in other cell types. So how does gene editing work at its core? Again, I'm massively oversimplifying here, but the key enabling feature is the fact that in eukaryotic cells, a DNA double-strand break is actually not often a lethal event. We suffer DNA damage, various kinds of lesions pervasively all the time. I mean, I think the numbers are thousands per cell per second in rapidly dividing cells. So we possess a multitude of different DNA repair pathways that need to rapidly recognize breaks and repair those breaks. And so gene editing really combines the ability to make a targeted double-strand break with the ability to, at some level, control the way that the cell repairs that double-strand break. So in the kind of simplest embodiment, you could imagine combining a double-strand break with an exogenous repair template to guide the cell to insert this new sequence through homology-directed repair. Again, this is probably not appropriate for this audience, but if we want to think about a simple analogy for gene editing, it's very comparable to find and replace in Word. You have a search term. In our case, that would be the guide RNA. You have a replace term. That might be that repair template. And we're going to carry out the same functionalities that you might do in Word, but with the language of the genome. But we need this to be exquisitely accurate because, of course, the genome in a human cell is some 3.2 billion base pairs. And if we want to really be able to harness this for treating disease, we need to be able to find, in some cases, a single letter mutation. So June 2012 was the landmark publication from Jennifer and Emmanuel's lab. It took about 6 months for the first descriptions of this core functionality being successfully applied in human cells for targeted genetic modification. And then in the years since then, there's really just been an explosion of studies using CRISPR in a variety of different cell types, human cells, human organoids, stem cells, but then, of course, a growing number of different model and non-model organisms. And I think it's because of this versatility, both in the types of cells where CRISPR is functional and the different ways that CRISPR can be delivered and used, has really led to this explosive growth in the number of studies that have adapted this technology. So again, in 2007, if we're just plotting the number of publications in PubMed that have CRISPR in either the title or the abstract, I was lucky enough to start working in this area in 2009 when you could read every paper in the world on CRISPR in about 3 days. Now, every day, there's some 25 or so publications that get popped into my inbox. 2007 was that landmark paper in Yogurt. 2012 was the first description of how CRISPR-Cas9 functions as a DNA cutting enzyme. And then I haven't actually continued this plot out to 2020, but I think this exponential growth has continued. So again, CRISPR is not the first gene editing technology, but it's by far the most easy to use. And I think that's why there's been this massive growth. Importantly, the onset of CRISPR technologies also kind of happened at the same time as companies like Adgene, which, if any of you are aware, is a non-for-profit plasmid repository based in Cambridge that essentially distributes plasmids to any academic researcher in the world at a nominal charge. So nowadays, a lab like mine, the minute we develop a new CRISPR technology, we send our plasmids to Adgene, and any researcher in the world can easily pull it up on a shopping cart and have it delivered next week by FedEx. So this has really completely eliminated the slow spread of information and reagents that I think 20 years ago you had to email the corresponding author. It might take a month for them to get back to you. Maybe they're too busy. Maybe they forgot your email. Nowadays, you can just have those reagents in your own lab essentially overnight. So this versatility has allowed CRISPR to rapidly infiltrate a number of different research areas. I'm not going to have time to go into many of them, but I'll just give you a couple snapshots before we come around to human therapeutics. So I think the first, which is often ignored or at least not given its enough attention by the media, is really that CRISPR has changed the way that most researchers do basic research. And that's because now we have just an incredible set of tools to perturb this basic relationship between a cell's or an organism's genotype and its phenotype by making targeted changes in any gene of interest, or in some cases, in every gene in the whole genome in a single experiment using multiplexing. Here's just a couple colorful examples of model organisms that have now become model organisms because of CRISPR's ability to now do genetics. So, the butterfly, what genes are involved in wing coloration, that's now been studied using CRISPR as a gene editing tool. This axolotl or Mexican salamander is an interesting model organism because it can completely regenerate amputated limbs, so it's a popular model system for studying stem cells and tissue regeneration. And the killifish is a very popular model organism for studies of aging. It's one of the shortest lived vertebrates that can be bred in captivity. And so now, with a new set of genetic tools to really study human-like aging-related diseases in killifish, it actually becomes tractable to do this at scale in a timely fashion. And then, of course, this is just a poster child for thousands of papers that have used CRISPR in human cells to understand the basic biology of disease. So in this particular study, which is from a number of years ago now, they wanted to understand what genes are involved in cancer metastasis. And so here they weren't using CRISPR to correct cancer driver mutations. They actually wanted to create more metastatic tumors using gene editing, but doing so in a whole genome-wide experiment. So the experiment here, they took a cancer cell line, they edited it with a library of guide RNAs so that in a single dish of about a couple 100 million cells, every gene had received a mutation, but only one mutation or one edit per cell. And the hypothesis was some edits will make those cancer cells more metastatic. They then took those cells, injected a mouse, and now collected metastatic tumors from distal tissues, and then could deep sequence, use next generation sequencing, to go back and figure out which guide RNAs, and therefore which genes, when edited, led to an increase in metastasis formation. So again, we're not targeting cancer using CRISPR here, we're understanding basic genetics of what causes this cancer to become metastatic. Just one brief slide on agriculture. I know it's not the focus here, but there's been a lot of excitement from the agricultural community to adopt CRISPR as a new way of making targeted modifications in major crops. I think there's going to be only more controversy coming here. What CRISPR is being used to do is make targeted changes without introducing foreign DNA. So based on how you define a GMO, these products may or may not be a GMO, and I think these are probably the first CRISPR products that are going to end up in broader society in the marketplace. Okay, and then finally, coming to human therapeutics, I think for many of us in the field, this has really been the dream for the development of CRISPR is, can we leverage this amazing power to target and potentially cure diseases by removing or eradicating genetic mutations? And we'd like to be able to use CRISPR not only to repair mutations, but actually address deletions or insertions in DNA. And then separately from really targeting genetic disease, there's a whole other growing area of therapy combining CRISPR with immunotherapy to take gene editing and improve the ability to generate CAR T cells, for example, that are combined with checkpoint inhibitors and things like that to really target cancer in this new way. So this is just a very small sampling of some proof of concept studies. I wanted to make the point here that growing cells in a petri dish, patient-derived iPSCs, organoids, it's become quite trivial to repair a variety of different types of mutations in cultured cells in the laboratory. Cystic fibrosis, Fragile X, hemophilia. The bottleneck is no longer really how to make the edit inside of a cell. It's really becoming how to deliver these modalities into organisms in a way that's safe and efficient, and how to control some of the off-target effects, things that I'll talk about in a couple minutes. There's also been a number of studies that have kind of shown very promising data delivering CRISPR in vivo into either mice or more recently for Duchenne muscular dystrophy into canine. So there's a lot of progress being made, but really I think delivery is time and time again really the kind of the gatekeeper, the bottleneck for how to realize a lot of the promise of this technology. This is just a summary of the two major categories where we could think about how to deliver CRISPR into patients. At the bottom, you have in vivo delivery, where you might systemically deliver CRISPR reagents, perhaps encoded in viral vectors into the blood, or perhaps to a targeted tissue type. What's been more quickly developed, because it's lower hanging fruit in the clinic, is ex vivo therapies, where you can remove a patient's cells, for example, hematopoietic stem cells or T cells, modify them in the laboratory with CRISPR, where you can now do very extensive quality control, looking for off-target effects, things like that, before transplanting these corrected cells back into the patient. This story, I think, was kind of one of the first really exciting indications that the combination of gene editing with immunotherapy could change the landscape of cancer treatment. This story is a couple years old, and it actually didn't use CRISPR. It used an earlier gene editing technology called TALENs. But nevertheless, I think it was a really exciting case where a girl who was dying of leukemia was approved for an experimental dose of gene-edited T cells on a compassionate use basis because she happened to be getting treated in the same hospital where this research was taking place. And that ended up sending her cancer into remission. And that work's since been published. I don't know why the references are so low resolution. So slight updates. So there's been now a number of clinical trials going on for cancer since 2018 in China. And just very recently, a couple weeks ago, there was some reports from a study led by Carl June at University of Pennsylvania showing in just a phase 1 trial that at least the delivery of CRISPR-modified T cells in 3 cancer patients was so far safe. Again, this was not reporting on efficacy yet, just no adverse safety downsides. And then the first sickle cell program has been going on now for about a year. And again, here there was some very exciting early data from the very end of 2019, where I think 2 patients in the US were treated for sickle cell disease using an ex vivo therapy, and there was a very nice story in NPR on one of the patients shown here in the end of December. So again, I think it's too early to say if these are going to be the panacea that we hope for, but I think we're kind of right at this junction when the next couple years are going to be very telling about the promise of these types of therapeutic approaches. And these are some more of the indications that are currently being pursued by some of the companies working in this space. So, separately from delivery, I just wanted to briefly touch on some of the other major areas where I think as researchers we need to really focus our attention. I'm not going to go through all of them, but I wanted to just briefly talk about DNA repair and off-targets. So the first, I told you earlier that this was massively oversimplified, and that is indeed the case, because a cell can repair a double-strand break in many different ways, and that repair is actually quite heterogeneous. So that's been a real problem for researchers because we really can't control exactly what repair is made. The best we can hope for is that across a population of cells, hopefully enough of them were repaired in a manner that is therapeutically significant. This just gives you a slightly less oversimplified version of these different repair pathways. This end joining pathway is actually the most efficient, but it's often the one that is least useful for doing precise modification. So in the field, it's really been a major goal to increase the efficiency of homologous recombination using an exogenous repair template, but this is really still one of the major bottlenecks for controlling how a double-strand break gets repaired. And maybe I'll just stress the point one last time here. CRISPR is not actually doing editing. The way that it's, and there's caveats there because there are new CRISPR tools coming out every month. So there are recent tools, if some of you heard about prime editing, where you can actually, to some extent, do editing with the modified version of Cas9. But the way that most of the world uses CRISPR-Cas9 for gene editing, the only thing CRISPR-Cas9 is doing is making a double-strand break, making a lesion. The cell is repairing that break to end up with an edited allele. And this repair process, you're at the whim of the cell. So if a cell doesn't make the right protein factors to catalyze this recombination pathway, then you're out of luck. So that becomes a really important concern if you're working in post-mitotic cells, like neurons, where the cells actually aren't even expressing the necessary enzymes to use this pathway. Another area that's gotten a lot of attention is how to control or address the so-called problem of off-target effects. So we have a nuclease, it makes DNA breaks. It uses a guide RNA to identify target DNA sequences using RNA-DNA base pairing, but the genome has 3 billion potential target sites, and with a 20-letter query sequence, inevitably there will be highly similar off-target sequences that might have 15 of the same 20 letters, or 16, or 17. So it was appreciated very early on that you can see the evidence of off-target mutagenesis, where partially matching sequences in the genome are errantly targeted and modified by CRISPR-Cas9. So obviously, if we want to deploy Cas9 as a therapeutic modality, but in the same cells where you're modifying beta hemoglobin, you're also introducing a mutation in other genes, that's obviously going to be a problem. So this has been another major area of attention, is both how to understand where are off-target effects being observed, what kinds of molecular features in Cas9 can we exploit to mitigate those off-target effects, and then even this study got a lot of attention 2 years ago because even at the right on-target site, there can be kind of large deletions or chromosomal translocations. So I think we've gone in the last 5 years from a point where any edit with CRISPR is amazing to now we want to get to the point of precision where we're really making only a single change and we can use genome-wide sequencing or other kinds of profiling methods to really ensure that across a therapeutically relevant population of cells that might be injected into a patient, that we know that none of them have undergone a mutation that might be oncogenic. I won't talk about these, but efficiency continues to be a problem. Often the precise changes we want are a low percentage of cells that are modified, and in some disease indications you just need higher numbers to be therapeutically relevant, so that's an area where we need to improve. And then an interesting development is in the last couple of years, multiple groups showed that Cas9 comes from, the Cas9 that many people use for gene editing comes from a skin bacterium, Streptococcus pyogenes. And so they set out to test whether or not humans have pre-existing antibodies against the Cas9 being used in gene editing and being used in these therapeutic programs. And that was in fact found to be the case. So there's now also been the development of either going to bacterial orthologs, related Cas9 genes that come from bacteria that don't see humans, so we wouldn't have a pre-existing immune response, or using protein engineering to mitigate any type of immune response. Okay, a few slides on germline gene editing. So I think it was appreciated pretty early on that with this powerful new ability to make targeted modifications, of course we can talk about using them in adult or kind of children patients, but given that there's been a whole industry set up to manipulate cells at the earliest stage of development, could CRISPR also be used in the context of reproduction? And of course this would, I often have to define the term germline to different audiences, I don't need to do that here, but this would not only affect the cells in that organism, but would pass on any changes to later generations. So I was actually part of a group of scientists and ethicists that met in 2015 to really begin discussing some of these questions over would this be possible? What kinds of guidelines should be proposed to regulate or to inform whether or not this should be a direction that physicians take? So in 2015, there were a couple white position papers that were published. The US government, at the same time, there was a note from the White House. The NIH will not currently fund any research that involves genetic modification of human embryos. And actually, even the US intelligence community was putting out statements about gene editing because of some of these big questions. Now, the NIH and the White House specifically mentioned clinical purposes, but in fact, in the same year, there was the first study from a group in China using CRISPR in the context of non-viable tripronuclear zygotes, showing that you could in fact make targeted genetic modifications in human embryos. The first study published out of the US came from Oregon, from Shukrat Mitalipov's lab. Here, the first study actually, it got a lot of attention, but it was frankly not a very well-designed study. They saw a lot of off-target effects. Their efficiencies were very low. In this study, what the Mitalipov lab did is they actually developed new ways of delivering CRISPR, where they co-injected Cas9 RNA protein complexes directly into the oocyte together with sperm, and showed that you can actually achieve much higher efficiencies as well as mitigate off-target effects because they were using a formulation of Cas9 that had much shorter expression, and so they had less off-target cutting because of that. So I think this clearly set a path in motion that, as probably most of you know, led to this news item from now over a year ago. This was announced just on the eve of the second international meeting on human gene editing that took place in Hong Kong, where Dr. Junghui He announced his experiments, his successful experiments to use CRISPR in human embryos that were later carried to term and resulted in the birth of 2 twin girls. I don't have time to talk about exactly what the experiment was that he did, but obviously this got a lot of attention, so maybe there'll be a time for a later discussion. I don't want to be too cursory in going through this, but I just did want to update you on where things are at. So I think many people in the science community think not only was this highly unethical for a lot of reasons on the question of was this medically necessary, but frankly the way the experiment was done, he was introducing edits that had never been seen in an animal model, much less any human patient, and yet these were still used clinically where we don't know what the phenotype from those edits will be. So actually, in the end of December, just a few months ago, he was sentenced to 3 years in prison, and this was after there was a lot of public condemnation of his work, and he was under house arrest for some time. So, yeah, certainly a very dramatic evolution in the germline gene editing area. There have been calls now on how do we move forward. I mean, the technology's there. There's questions about are there medical needs where this would be justified? If so, what would they be? Who should be making decisions about that? So I don't have answers, but we can have a discussion later. There have been calls from the World Health Organization to kind of set up a global registry to at least make sure that any clinical trials, whether it's in somatic cells or germline cells, is described transparently and openly. And there have been a number of other calls for either a very strict moratorium or ongoing discussions on how to really reach some kind of consensus internationally. So I always tell people, I think these are real issues that need to be addressed. It shouldn't tarnish the possibilities for using CRISPR in human patients for non-germline uses. So I think really we need to make sure we're openly discussing these to use CRISPR in the most responsible way. And I think, again, what I spend 99% of my time thinking about is how can we make sure to develop CRISPR technologies and tools that will make treating and potentially curing diseases in human patients a reality. Okay, last couple of minutes. These guys didn't invent CRISPR, or neither did Jennifer Doudna or Feng Zhang or George Church. It actually is nature that invented CRISPR, so I always like to end by stressing that. And I think we found time and time again that nature is the best engineer, the best inventor It's had billions of years to solve a lot of different complex problems. So kind of the lens or the thinking of my lab is to go back to this very interesting and pervasive genetic conflict between virus and bacterium to keep mining for new tools. And I'll just leave you with a couple short snippets. Number one, I told you at the beginning, Cas9 is the enzyme that's gotten all the attention in CRISPR, but it's a tiny, tiny sliver of the CRISPR diversity that exists in nature. So just like Darwin's tree of life, we have our own CRISPR tree of life. These are all the different spokes or branches of the CRISPR tree. There's now 2 classes, 6 major types, over 30 subtypes. This little sliver up here is Cas9. There have been now new technologies built up around Cas12, Cas13. As far as I know, Cas14 is the highest we've gotten, but there's actually other gene names that don't use Cas, so the true number is in the high number of dozens. And all of these have different molecular properties. They all use a guide RNA, so in that sense, they're all programmable. But the way that they recognize targeted nucleic acids and how they cleave them is different. And in many cases, those differences can be the inspiration for really augmenting this CRISPR Swiss Army knife or this CRISPR toolkit with all these new types of functionalities. So really quick, an area that my lab has been really interested in just the last 2 years since I got started at Columbia, we actually discovered something really interesting, which is there are transposons or jumping genes that exist naturally that have CRISPR-Cas systems jammed inside of them. And what we found is that these transposons encode enzymes called transposases that can be programmed with CRISPR, programmed with guide RNA. I'm going to gloss over this just in the interest of time. For those of you that remember learning about transposons, these are mobile genes or selfish genes that can pop themselves out of one site and splice themselves into a new site. So this cutting and pasting functionality has actually been a huge boon for a lot of different technologies. Genetic screening, doing DNA barcoding, it's actually used in gene therapy. But a downside for all these tools is that the enzyme that does this DNA splicing or DNA insertion is totally random. It doesn't have any rules for where it inserts DNA. And so that means that for gene therapy, you can insert or splice a gene into a cell of interest, but you don't know where it's going to go, just like a lentiviral vector. And that has some real downsides because we want more control over the site of an insertion. And so what we found and published last year was that with these transposon-encoded CRISPR systems, you can use an RNA guide for making very precise DNA integration. If we look at how this compares to Cas9 in the context of gene editing, let's say you want to put in a transgene like a CAR for cancer immunotherapy. With Cas9, you make a double-strand break, and then you hope and pray that the cell will use your repair template to repair that break. But most of the time, this gets repaired a different way. With our technology, we now have a completely self-contained enzymatic system that not only targets the right place, but does all of the chemistry to insert the gene of interest into that site. There's no DNA break, which means there's none of those off-target effects. There's none of those heterogeneous repair outcomes. You get this very clean insertion, and we published a paper describing this in great detail. So again, we're going to eliminate all of these different forks in the road for a double-strand break and deliver a technology that can make a precise insertion. In my lab, I'm a biochemist by training. We think a lot about structure-function type questions. How do we understand what these machineries look like so that we can engineer them better? So earlier this month, we published our second study together with Israel Fernandez, where we solved cryo-electron microscopy structures of exactly this machinery right here. It's a beautiful structure. What you're looking at here in red is the guide RNA snaking along the length of this protein complex, and then colored in the different colors are the different protein components, which essentially form a filament along the length of the guide RNA to catalyze the process of DNA targeting, where you need to unwind DNA to make those RNA-DNA base pairs for identifying target sites. Okay, hopefully I haven't lost you. So It's a pleasure to come over to the east side, which I don't do all that often. I'm up here at Columbia Medical Center in the Hammer building. This is my nascent research group, a fantastic group of students and postdocs. Thanks again to Hannah and the other folks here for the invitation, and I'll be really happy to take some questions.
First of all, thanks for bringing some of us into the newest world. My question is not even modest, it's a little grandiose, I think. We're about to have, some of us believe we're about to have a pandemic. May or may not be true, but I believe we're about to have a pandemic. So in that way, how would you use CRISPR to try to stem the pandemic?
So I don't see an obvious approach to use CRISPR to directly prevent the pandemic, but I can tell you something that's actually happening. I don't think I even have it on here. There are two companies that have been founded in the last year and a half using CRISPR tools for molecular diagnostics. So it would take me a little too long to explain the details, but they are using CRISPR enzymes that can detect a given sequence of RNA or DNA with high specificity, and they've transformed that into a very high sensitivity rapid readout. And what they're trying to do is build a kind of point of care chip that you could deploy all around the world, where without any expensive technologies, no thermal cyclers, no PCR, no need for equipment, you could detect diagnostic signatures of a given viral or bacterial pathogen to at least inform things like containment or quarantine. So both of these companies recently released white papers on how they've already developed tools, specifically coronavirus, with their platforms. Now, again, that's not going to address the disease, but it could at least help with detection and rapid diagnostics.
Somewhere in the middle, you talked about off-target effects that may occur, and in fact, a couple of the earliest humans who had this technology actually developed leukemia. I'm wondering if you were to do the procedure in vitro and then grow organoids, would the fact that you're getting multiple replications make it easier to find when there was an off-target event, and especially maybe if you had a jumping gene, which you would only recognize if it started replicating?
And with the leukemia example, are you referring to the gene therapy work from the early 2000s, I'm guessing?
Well, that's the first one I was talking about.
So that was not CRISPR, of course. That was using viral vectors. And I think that does highlight a major risk of a vector that inherently inserts transgenes in a random fashion. You can't control where it goes. So that onset of leukemia was because of this insertional oncogenesis. So hopefully a technology like this transposon technology would give you more control over where it goes. But you bring up an excellent point, which is how would you ensure that, let's say, an edited cell population doesn't have that 1 in a million that could become oncogenic? So previously what was often done or expected is just that you put the cells in a mouse and you see if they grow tumors. I think now we can do much better, which is we can not only do that or look for growing organoids in the lab if you see any evidence of some transformation. We can also do this genome-wide analysis. There's a number of different molecular tools and approaches that have been developed to use next-generation sequencing to really query in an unbiased fashion any off-target mutation, any off-target insertion or translocation. So I think you're absolutely right. It's going to need to be a combination of passaging cells extensively in vitro, looking for transformation in animal models, but also using these next generation sequencing approaches to actually look at the genotype level if there's anything that is awry. Yes?
Your nature paper on the CRISPR is really a great technology. I was, I think your nature paper is really exciting. I was wondering, since the nature of the transposon is to be able to jump genes, how stable is it after it's inserted with this CRISPR technology? Is it able to like subsequently move and then your inserted gene is now non-functional?
Yeah, it's a fantastic question. So we're still developing this in human cells, so the work we've published so far is in bacteria, but you're absolutely right. The question is, if you're going to use this even in bacteria for other things or in a clinical setting in human cells, how do you control that it gets in once but it doesn't keep jumping? And the strategy we've taken is to separate the expression of the transposase from the mobile DNA. So if you deliver the transposase in a short-acting formulation, like if you transfect protein, purified protein, it will only have a very limited window of time in which to act before it'll be naturally recycled in the cell. And then the DNA that we're inserting doesn't actually encode the transposase. So in nature, these obviously jump as autonomous units because the mobile DNA encodes the gene that mobilizes it. But when we use this, we can now carve all those genes out, deliver them in a different way, and the DNA that we insert has just the little ends that the transposase recognizes, but we can clone any transgene inside we want and ensure that we don't have the transposase around after that first insertion.
I have a question. So where do you think we'll see the first FDA-approved CRISPR application? What's in the lead right now?
Again, outside of my expertise, but I think based on the recent news and the initial reports, I think probably ex vivo CAR T-based therapies seems most likely, especially because it's relying on, but the news from the sickle cell program is also very promising. I would have to say probably one of those two, partly because both are resting on the foundation of, in the case of CAR T cells, other clinically and FDA-approved treatments. And for sickle cell, we already have a very long history of using bone marrow transplants and using hematopoietic stem cells from a donor into a recipient. So all you're doing is trying to recreate that with the recipient's own cells that have been repaired. But a lot of the other questions of conditioning and that kind of thing, I think you can rely on a lot of the work that's already been done. So that'd be my guess, but again, I really am not an expert on that. There's maybe one more question in the back.
Isn't the most unethical thing about the Chinese editing that they did it first?
What was the question?
If the most unethical thing about the Chinese germline editing experiment is that they did it first. I should have made more clear I was quoting you instead of saying it myself. I mean, this is something that there's any number of articles that have posited, are we setting up an East versus West type of conflict, and is there now going to be a race to harness these tools for generating superhumans? I don't know if that's kind of the tack you're going at, or I kind of agree with you. I think there are probably some scientists that secretly feel a little bit maybe envious that the regulations apparently are more lax in other countries, although, I mean, he's in jail now. I don't know if there is there was a thought out there, maybe there was some implicit approval for his experiment until the rest of the world said, what a travesty, and then they felt like they had to respond by showing publicly that they were going to punish him for it. Yeah, I don't know if you want to say any further comments, because I think that's an interesting part of all this is China has been a leader in a lot of the work.
So I was in the church life at the time when this was happening, and there was a discourse around people feeling like they did it first, and a lot of the conversation was about that, as opposed to, this is so unethical, or what about the off-target effects and all this sort of stuff. There is that sort of conversation around.
I think it was medically unindicated, though. I think in that case, it was a gene therapy to prevent infection with HIV, for which we have excellent therapies. So we can prevent the maternal fetal transmission of HIV with drugs. So it was medically unindicated. Plus, it's dangerous.
But I think in the history of innovation, like anything that's ever come out, we know. I think Jennifer Doudna spoke about this. It eventually is used for indications which we would otherwise think unethical. So that then, when you take that as your foundational concept, then why regard it as unethical? I think that maybe gets a bit philosophical.
Yeah, I mean, I didn't mean to give a too superficial overview, and I don't actually find the idea of germline gene editing as immoral or unethical in and of itself. My issues personally are mostly to deal with access and equity in the way that something like that would be developed and offered, and because that's already a problem with assisted reproductive technologies, which tend to be not covered by insurance. And if you have $20,000, great, you can use IVF and pre-implantation genetic testing. If you don't, then you can't, and you're infertile or you have fertility problems, then you're out of luck. So I think my personal concern is, how does this just accentuate some of those inequalities? But I agree. I think we could have a longer chat about this, but certainly there's a lot of different sides to this, and there has been a large backlash because I think it's safest in a public spotlight to be very critical across the board, but there is a question, like, in the next 1,000 years, are we really not going to do germline gene editing? I mean, I don't know.
Okay, well, let's thank Dr. Sternberg.